Welcome to Top Notch Farming. SaskCanola has partnered with Alberta Canola, Manitoba Canola Growers Association to deliver these presentations to you. My name is Keith Fournier. I'm a Director and Research Chair of SaskCanola. I'm happy to be chairing today's webinar. We are glad to have you joining us today for the first of 4 webinars in the 2021 Top Notch Farming webinar series. We certainly miss being able to see you in person this year, but are thankful for the technology we have available that allows us to come directly to you with useful information for the upcoming growing season. The 4 webinars in this series cover a wide range of topics, including Blackleg and Verticillium Stripe management today, clubroot management on February the 11th, harvest and storage management February the 18th, and soil management March the 4th. We invite everyone to join all 4 webinars. A few housekeeping items before the presentation. Continuing education credits are available for certified crop advisors and certified crop science consultants. To earn credits, please send us your full name, CCA, CCSC number in the chat box. We will fill in the sign-in sheet and send it to the appropriate person. Another option is to scan the QR code using the CCA app. The QR code will be displayed at the end of the last presentation. If anyone has any questions during the presentation, you may type them into the Q&A box in the right-hand corner of the control panel. There will be a 30-minute Q&A session at the end of the presentation, and we'll be sure to answer your question. Please use the like button beside the question. Questions with more likes will be addressed first. During the presentation, you will be muted so that there will be no feedback that will interfere with the audio. We encourage you to use the chat box and to add your discussion, to add to the discussion of each topic. So why don't we start right now? Tell us your name and where you farm or work. This webinar will be recorded and the link will be emailed to you at a later date. I am pleased to introduce our first speaker, Dr. Dilantha Fernando. Dr. Fernando is a professor at the University of Manitoba. His research focuses mainly on plant pathology, specifically epidemiology and genetics of host pathogen interactions. He's going to give us a blackleg research update in the next 30 minutes. Thank you so much for the presentation.
Thank you, Keith. Good morning, everyone. I'm going to first see whether I can share the screen here. Okay. And okay, so once again, good morning, everyone. It's a real pleasure to be invited by the SaskCanola. Thank you for that. I'm glad that I'm able to make this presentation on the blackleg management. And the reason that I say that is blackleg has become the most important disease that we as farmers need to tackle. And also Keith mentioned a very important point. I hope this presentation leads you to a better management strategy in the coming year and the years to come. So to do that, the question and the title that I have here is Blackleg Management. Are we in this together? So keep that in mind. Are we in this together? So if you are not familiar with the disease itself, I just wanted to give you a bit of a background of blackleg. So blackleg, the first thing that you need to know is that blackleg affects all parts of the plant, from the leaves, the seedling leaves, the cotyledons, the stems, and then subsequently, when the disease has had an impact, there will be lodging and the plants will die and there will be no seed set. So why is CAP funding this the work that I'm going to be talking to you about? Predominantly, it is because of maintaining the canola supply and the trade that we do with other countries. As a player who has a huge stake in the export market, Canada has to take into account what other countries are looking for when we sell our seed and oil to other countries. So keep that in mind as well. So I think the best way to introduce my presentation today is to bring you to the most talked about thing today, that is the COVID-19. So I ask the question again, are we in this together? So look at the right panel here. This young girl is doing the right thing by wearing a mask. This lady on the left is fighting, saying that she doesn't want to wear a mask. There are people who are here on the middle panel who are congregating together. And here people are doing the right social distancing. So if the social distancing was done correctly, there is a 75% reduction of that disease within 30 days. Whereas when there's no social distancing, there's an extra 408 infected people in that population. So the other question that I have for you is, what's your excuse for not wearing a mask? Okay, so keep those in mind as we move along. Now, a good example is from Manitoba itself, where I am. Somewhere in November, there was a surge in the disease, which had been managed so well in the first few months of the first phase of COVID. And then there was a huge surge, and we were put into a code red in Manitoba, which meant that there were proper social distancing and mask wearing implemented, and also there were other restrictions that came along. Look at what has happened in from that period to January. There has been a very steady drop of the COVID-19 in Manitoba. So we learned lessons from other epidemiological situations. Here's a human tragedy, and now we can take it to the Canadian importance of our canola, the darling crop of the farmers. So this is a project that was funded in 2018 when COVID was not there. But I think the scientists were thinking on the right lines and trying to understand, how do we improve blackleg resistance durability through our gene rotation in commercial fields on the Canadian prairies? And we wanted a science-based stewardship program to be implemented. So the SaskCanola and other grow organizations, along with the Canadian government, came along to fund this project at $1 million over 5 years. So we are into the third year, and we have a lot of information to now give back to the farmer who has funded this work. So the project is led by myself, and I work for Manitoba. Dr. Gary Peng is working for the Saskatchewan province, and Ralph Lang for Alberta. So now look at this epidemiological information. Just like the COVID, we are seeing prevalence of increase in the prevalence of this disease in almost all 3 provinces. And if you look at even the disease incidence, the disease has been increasing fairly well in the last 5 to 7 years. So these are important things that we need to understand, because that leads to certain issues with yield for the farmer and the trade for the industry and for Canada. So you can see on the left-hand panel here, Mr. Justin Trudeau, our Prime Minister, touting Canada's agricultural safety amid China's canola dispute. So that canola dispute came along with because of Blackleg, and we have been systematically working through diplomatic channels and through science to bring about the disease to a level that the countries that do trade with us will accept our seed. And the other aspect is the yield loss. Yield losses cannot continue because it hits the bottom line of the farmer. So going back a little bit, in the first phase of COVID, we had a first phase of looking at what can we do for the farmer to bring about a mitigation of blackleg. So this started in 2011, and my PhD student, Sheau-Fang Hwang, started to look at what was really available in the Canadian germplasm of Canadian Brassica napus, what kind of R genes were there. So this was the first time that R genes were studied in Canada. And we were able to successfully show that there are several R genes that are available, but the predominant one was the RLM3 gene in our canola varieties, whether it was BASF, Bayer, Monsanto, or Cargill, whatever company, the predominant gene was RLM3. Now, at the same time, another student in my lab, Zachariah Liban, was looking at the pathogen population, trying to understand what we had as the pathogen population in the prairies. And I bring your attention to this right here, where my cursor is, where you see that the orange shows the virulent form of the AVRLM3, which means 97% of the isolates that are in the field are virulent, and only 3% are avirulent. With the avirulence matching the R gene, that's the only time that there will be no disease. In other situations, when there's virulence, 97% of the time, even when there's an R gene, there will be disease. So now you can bring these two together and understand why we were starting to see in that graph that I shared with you earlier, there was a steady increase of the disease in all three provinces, and that was the reason for that, because predominantly we had RLM3 in our varieties, and predominantly we had the virulent isolates in the field. So even though it was an R gene, it was not acting as an R gene. So that was the success story of the first phase. So we were able to understand what R genes are there, and we were able to understand what pathogen populations are there in the fields of the farmers, and then suggest R gene labeling should come along to be adapted in Canada. And that was approved by the Blackleg Steering Committee, and then taken up by the Canola Council of Canada. And you know, in 2017, this was formulated to be implemented. So what led to these particular publications that I have listed for you. Now, coming back to how we normally look at the blackleg resistance, there was the one that WCCRRC has been using in their screening, that is to label a variety as R, MR, MS, or S. Now, once you do that, you immediately see that that is not exactly telling you what R gene is being carried in what variety. So the new labels of major seedling resistance genes, that's the R genes, really help to propel what has been used by the WCCRRC. So here's the WCCRRC's way of resistance identification. Anything that is called R has to have a percentage of Vista check of 0 to 29.9, which means that it is far less in disease, and that is why it is called an R. And you go along with that in the testing protocol. So if this is how you see it, it will be rated as what it is, and then get a R, MR, MS, or S. Now, the new Blackleg major gene labels, as I said, propels that because the farmer comes to know what gene is there in the variety that he or she is going to buy. So, for example, if a cultivar alpha is labeled R, but with a parenthesis BC, B stands for the resistance group B carrying RLM2 gene, and C RLM3 gene. So now the farmer knows that there are 2 genes to protect his crop or her crop, and that if that matches well with the field's pathogen population, then they can go with it. But in addition, because of that, they will be now able to select varieties that are going to work in the long run, rather than grow the same R gene again when there has been disease, high disease in the year that the canola was grown. It is recommended highly that you don't grow that because it's called a bad rotation. And especially if you are in tight rotations with canola, that is a very important part to be remembered. So good rotations are any other R gene that you can find other than the R gene that was used in the previous year. So this is a fine example from Australia showing what I'm saying. So both these panels, left and right, has the same pathogen population. That is the pathogen population coming from group D. So group D pathogen population is in both. Now here, group D, there was another variety grown that was group D, and you can see quickly how the pathogen was able to overcome the crop and cause disease. Whereas here, the pathogen was not able to cause disease, and the plants grew very well because it was a different group, a different R gene that was grown in the Eyre Peninsula in Australia. Now, we have a bigger advantage. The Canadian advantage is, as farmers, you are able to come to know what pathogen population you have because we have developed CASP markers to identify this. So CASP markers, if you send your samples to a seed testing laboratory like Discovery Seed 2020 or PSI, they will be able to ID the races of the blackleg pathogen population and tell you, please do not grow this particular R gene, but we recommend this particular R gene to be grown. In a few slides later, you will start to see what I mean. Now, along came at the same time, I think in 2018, the first, if I am correct, the first to label the R genes was Monsanto at the time, now Bayer, a decarb variety. And look at what they have here, R with parenthesis ACG. A stands for RLM1 gene, C for RLM3, and G for RLMS. So once the farmer knows that, the farmer is at a very high advantage to select the right variety. Now, when you think about selecting, there are other things that come into play as well. You look at the herbicide resistance, yield potential, cost, availability. But if at harvest you have seen that there was blackleg infection and there was lots of blackleg in your field, then the opportunity is now there for you to choose the right R-rated variety that would match for your situation in your field, because the seed testing laboratory will give you a certificate with the pathogen races. You take it to the agronomist or the supplier who tells you the variety to be selected. And then you have selected the right variety. So these, we are very happy that the first phase has been a successful phase where now the companies have started to put the labels on. Remember, I told you earlier, it's like putting the mask on. We are doing the right thing. So data moving from a lab or greenhouse to recommendations to use on the farm is the most gratifying thing for a researcher, because if that is not done, data by itself is useless unless you can turn it into the recommendations to use on the farm. So this is the mantra that I use always when I think or train my students, that the bottom line of what we do has to be impacting the growers and the industry. So when we started the second phase, we started to look at what really would work and not. And here's a very good example from 2018. When RLM3 gene is grown on RLM3, this is what happens. A complete devastation and blackleg infection, whereas in that same stubble, RLM3, we have grown RLM4 and excellent healthy varieties are there. So this leads to the second phase of the experiments that I just told you earlier, to look at how successful these R gene rotations will be in grower fields. So to do that, I have Justine Cornelsen, who will be a speaker today, and also most of you as farmers know Justine, who works for Canola Council of Canada, as well as she's a master's student in my lab, and Dr. Jongwei Zhou, one of my research associates, are leading this project. So here's just to get you started, I wanted to give you a snapshot of what to expect. Here's two varieties that are labeled R or resistant. On the left-hand side, you see perfect, the variety performing to its name R. Whereas on the right-hand side, the variety has not performed well and stood to its name as carrying the R gene. So when you look at these in real field situations, you start to see that when you don't have a labeling, or when you have the R gene like the RLM3, which is predominantly grown, and when the pathogen population is predominantly able to cause disease, you have an increase in disease in your field, whereas the disease is reduced when you have ACG, where that is there are three genes that are in there. We will look at why that is later. So here, another field in Saskatchewan, you can easily see that because the AVRLM4, the avirulent gene of AVRLM4 is predominantly available, the pathogen has a lock and key mechanism with the R gene, and that is the E, that is the RLM4, giving protection even when RLM3 was there. So whenever there was only RLM3, there was disease, and if there was no labeling, there was disease. So I wanted to bring back this study that was done by Sakraya Liban, and you can see why RLM4 is still doing very well, because we have a very high population of the AVRLM4, that is the avirulent and not the virulent form. So in our work leading to the phase 2, we have gone and looked at scouting fields and using 2020 labs to do the PCR profiling using CASP markers. So I'll go through these very quickly because there are several fields that I wanted to show. And here's Nobleford, Alberta, showing on 45M35 Roundup Ready canola, the rating that has been given by WCCRRC is a MAR, and you can see that there is disease. Now, keeping in mind, this doesn't have a R gene labeled. And even though there was good rotation, wheat, and then oats in 2019, and came back to 2020 with canola. The pathogen was able to overcome. And you can see that there was both the sclerotinia and blackleg in that particular field. But I am focusing on the blackleg where it was diseased. And you can see when you take the stem cuttings, you can see easily how blackleg was impacting that particular field. Now, when we send the isolates from the stubble to the 2020, this is what they came up with. The genotype shows AVRLM3, but the phenotype that is predicted does not, because the AVRLM4 and 7 are present with the 3. The 3 is masked. The 3's activity is almost non-existent. So there is no match, and the pathogen is able to overcome and cause disease. So now a few other fields. This is another variety, and this variety was labeled R, L233P, and you can see the amount of disease in these plants, and that is definitely not acting like a resistant variety. And again, once we do the profiling, the PCR shows that AVRLM3 is there, but the predicted PCR phenotype is that the pathogen does not have a ability to have the AVRLM3 active because of the presence of AVRLM47. So here's a few more in Coaldale. Again, you'll see the disease. What I wanted to emphasize here was that the years leading to that 2020 has been very carefully used with different different crops, but yet the pathogen was able to cause disease. Now, this is a little bit more tighter rotations because 2018 canola was grown, and again, it's 45 and 35. You can see how much of disease there is. There's another field in McGrath, Alberta, the Liberty Link variety, and you can again see the blackleg. So I'm going a little bit faster here just to let you know that wherever we looked at, if even with good rotations, here's 2017 canola, 2018 wheat, 2019 lentils, and 2020 the canola, still there was disease. So here's another in foremost Alberta. And that brings me to the take-home message in this part of my presentation. So if one company labels our genes and helps the farmer, and the other company is not labeling, this blackleg disease will keep staying in our Canadian fields, as I just showed you with many fields. That is not helping the farmer. So it is like some wearing a mask, as it is the right thing to do, and the others not wearing a mask, that will keep the disease spreading, the COVID disease spreading. So here's putting it back on our situation of our genes. The girl who's wearing the mask is like labeling our genes to support the farmer. The woman on the left-hand side who's shouting and saying that I'm not going to wear a mask is like not labeling our genes. So my question again is, what's your excuse for not labeling the our gene? Because we as an industry have to move forward together. So are we in this together? That's the question. So there's a second part of this work that we are doing, and that is to look at whether we can systematically rotate certain R genes in Manitoba, Alberta, and Saskatchewan to see how these R genes work in tandem in bringing about the disease to be low. Now, we have also introduced a new gene, RLM2, in 2020, And I just wanted to be very quick because I have a few other slides to share with you, just to show you that RLM2 was introduced to this rotation where, so for example, rotation 1, we don't have any rotation. It's the susceptible Vesta as a control. And here we have rotation 2 as RLM4 with no rotation, single gene, but no rotation. Then we have another rotation where maximum R gene diversity is looked at, RLM4, RLM3, and RLMX, another combination, then RLM2 and RLM4, and so on. So we had a strategic plan in all 3 provinces to look at this in different combinations of different R genes, major genes in combination. And to make a long story short, now this is new, fairly new information, so we will have to wait another year to see this. What you start to see is once you bring in a new gene, there is a reduction of the disease and the index of the disease in all 3 provinces when RLM2 was brought into the picture. So this is good news, excellent news, and hopefully that would be something that we will have to consider as we move along in the future to bring in more genes. And I know that companies are working very hard to do that for you. Now, one of the other things that I wanted to end my part of this presentation was with this thought. In the same field, you can have 2 varieties grown, one that was just with RLM3 and the other with RLM3 and RLM4. And you can see in the same field how different the reaction was. So what is important to remember is that you need to understand which R genes are going to be working for you in your farm. And that will not be your neighbor's farm, it should be your farm. How do you come to know that? By sending your samples to 2020 or Discovery Seeds or PSI to be tested to know the pathogen profile. So I have brought the COVID virus and the R gene comparison right on with one slide. So on the COVID side, we know the virus is transmitted from mouth and nose, sorry. We know on the blackleg side, the blackleg pathogen causes disease when it becomes virulent. On the COVID side, we know much about the virus and about the spike proteins that is highly talked about today. On the pathogen, on the canola side, we know much about the R genes in canola cultivars now. It was not there before 2013, but today we have that information. We know that wearing a mask and that will protect and help reduce disease. We now have shown that labeling R genes in cultivars protects and helps reduce disease. Now, on the COVID side, we know it reduces spread, and here we also know it reduces spread if we do the right thing. Now we have on the COVID side, vaccines to protect and reduce disease spread. I can say that we have R genes to protect and also Cas markers to identify the pathogen. So it's really a good opportunity. Now we reduce the economic impact and help businesses to thrive once again by reducing the COVID. And we do the same thing by reducing the economic impact and trade barriers and help the canola industry to thrive again. So what have we done? So if we think about the COVID-19 again, they have brought everything together, masks and social distancing, then rejuvenating the economy with stimulus packages. The vaccine development in several countries has helped, and then a global vaccination process for vaccinating all. Now, the same is true with our genes, bringing it all together. Comparing the incidence and severity data of L. maculans races causing the damage, validating the idea of our gene cultivar rotation within the field is a very important part. Information will help producers select cultivars, as I showed you, provide canola breeders with which major resistance genes to include, and that will lead to a lowering of blackleg disease severity across Western Canada that protects our canola crop and helps the industry and trade relationships. So that is the project that I am leading, and I have the pleasure of also talking very briefly about other projects that are led by other scientists who are doing some fantastic work. So Sheau-Fang, who is a speaker today, is leading this project looking at fine-tuning of the blackleg yield loss model in canola. So there's the team of hers that is mentioned there. And what they have done is to use different canola cultivars with different types of resistance from MR to R and compare with how, with a susceptible, how the disease will incur. And these are the canola hybrids that they have used. Here's the disease severity. And what is important here is when you compare with the control where there is no inoculum to the one with inoculum, you see a significant reduction in the yield, and that is consistent with any variety that was grown, moderately resistant to resistant. So again, it really corroborates with what I had shown already in farmers' fields. It doesn't matter whether it's R gene. That R gene will fail if we don't do the right thing. And here's the relationship. The way that the disease severity increase will have yield losses, and you can see how steadily the yield loss will increase, and disease severity close to 4 and 5 have the most significant yield losses that Dr. Sheau-Fang Hwang has shown. Now, another experiment under the CAP project is understanding critical window of infection for blackleg, led by Dr. Gary Peng from Agriculture and Agri-Food Canada. And here he has used different varieties again and compared it with Vesta to look at the disease incidence and severity and look at the window of disease going from inoculating at the cotyledon stage and later at the first leaf stage. I'll go right into that slide that shows when inoculated at the cotyledon stage, there's much more disease compared to the disease on the first, when inoculated on the first true leaf. And this is very evident from this as well. When the DNA was taken, the pathogen was present in much larger quantities and in most varieties compared to the first leaf stage when inoculated on the first leaf. Also, Dr. Gary Peng is leading another project on looking at adult plant resistance, or what we call quantitative resistance, because we spoke about resistance genes, are genes that are single genes, but there's also a need for us to enhance our understanding on the QR, or adult plant resistance. To do this, he has used a method called ddPCR. It's a quantification method of how much of DNA is present. To make a long story short, he has used 50 plus commercial varieties that carry R, MR, or S, and looked at how the quantification of the DNA will match with the R gene, the resistance of the line. And there's a very good correlation when you look at the ddPCR results along with the disease ratings in the field. It was a very good correlation. And that leads to show that petiole versus cotyledon inoculation also has a very good correlation when you look at the with the ddPCR, the DNA of the pathogen presence in the plant material. Now, there is another project that Dr. Gary Peng is leading that I'm part of, and that is looking at the interaction with the blackleg and the flea beetle, whether flea beetle damage leads to more blackleg infection. So here, that is because wounding, we have shown that when there is no wounding, there is no infection. When there is wounding, there is infection. And that relates to what happens with flea beetle damage as well. So what we have done in the greenhouse is to first understanding what wounding does, and we have been able to show that the critical period is the first 8 hours that the pathogen will be infecting. After 8 hours, the infection is less prone to cause disease. And these are field results. I'm just showing the results. They have not shown any significant outcome of what we have been expecting. So we are continuing with these experiments to look at whether we could get a better idea in the future. And these are also done in all 3 provinces. Now, Dr. Hossein Bohan, I'll end with Dr. Hossein Bohan's work, looking at developing tools for the rapid screening of canola germplasm, again for quantitative resistance or adult plant resistance. And what he has used is a variety castle that is there with the quantitative resistance and topaz that doesn't. And you can see the difference when there's some quantitative resistance. There's very good standing of the plant. Now, they have gone ahead and crossed that to get double haploid progenies, and there's a difference in the different progenies, and you can see a variety of QTLs or quantitative trait loci in when you do field experiments with these populations. Now, doing that in the field and in the greenhouse or growth chambers are shown to be fairly similar. So that is an important aspect for their research to move further. Nearly 50% of the population showed strong to intermediate resistance. So they have gone further to know that it is on the AO8 chromosomal location that these QTL markers are. So they are doing further work with that. And finally, they are also leading a project to look at further developing CASP markers to help the growers in identifying other R genes like RLM9. So to do that, they had used Vesta and used RLM9 to be transformed into Vesta. So when you transform the RLM9, the pathogen cannot cause disease when even in the presence of the AVRLM59, whereas Vesta is having the disease whether it is with or without. So you can see that the RLM9, when it is a transgenic plant, even though it is in Vesta, it is protecting the Vesta plant because the RLM gene is in there. So I wanted to take this opportunity to thank all these scientists who are doing excellent work, and my external collaborators, and also the work that has been helpful for us from the Australian and French groups that are also collaborating with us on numerous projects, and the many funders, including SaskCanola and other grower organizations. So that's my lab this year, and I would like to take any questions in the future as well. I have given you my email address here. So I'll stop there and take any, maybe I'll let Keith handle from here.
Thank you, Dr. Fernando. So yeah, please type your questions into the Q&A box. We will address them during the 30-minute Q&A session at the end of the last presentation. Also a reminder, if you are a CCA or CCSC, please send us your full name, your CCA or CCSC number in the chat box. Our second speaker is Dr. Sheau-Fang Hwang. She is a professor at the University of Alberta. Her area of research focuses on major diseases of canola, especially integrated management of soil-borne diseases. She'll be talking about Verticillium Stripe research.
Yeah. Share other quality.
Good morning, everybody. It is a very good honor and also appreciate the opportunity to give us a talk updates on the Verticillium Stripe research updates. And I think many farmers, you know me as a club roots person.
Share the other screen.
What?
You want to share the other screen? It's coming. You can just tell it.
From beginning. How to start beginning?
Yeah, so we continue.
I'm sorry, there's a little bit technical problem. And so many of you know me as a club roots person. And I've been working on club roots for 15 years. And so, tell the truth, today when I give this talk, I'm a little bit more nervous, just because I consider myself still is a very beginner to work on Verticillium Stripe on canola. And okay, so how did I get involved with the verticillium disease? As you can recall, the disease was the first time discovered in Manitoba in 2014. So then 2015, the CFIA got very nervous. And so, you know, immediately hired so many people running around, try to collect the soil samples. And then I was involved at that time when I worked with Alberta agriculture. So there's more than 1,000 soil samples were collected and then sent to Ottawa for PCR work. And then turned out about a quarter of them showing positive results. And also it turned out Manitoba get #1. I think at that time, probably 30–40% positive. And Alberta and the Saskatchewan, and not too bad. I think it's kind of like 11% range. Then the reason I got more involved with verticillium disease is because in 2016, the following year, and the farmer bring to my attention and to tell me he had some kind of verticillium disease. So that's why we went to his field, and we saw this in the field. As you can see, and the leaf chlorosis, the wilting, and the early ripening. And so you know, plant pathologists when see this typical disease always get very, very excited. And so I thought maybe I got a Verticillium longisporum. So I carry out some more research in the lab. So I take more pictures just to show you this is a wilting plant, and then also show the microsclerotia on the stem. Then also, you can see the whole vertical brown and appear on the stem. The tiny microsclerotia is there, and then inside the stem, you can also see this dark mycelium and the kind of small microsclerotia there. So if I go to the field back again in the same spots, and I will, and after swathing, and I will see these plants like that. But notice there are no shaded. Okay, but I just kind of showing the microsclerotia and the kind of dark color, gray color, all these. So I got excited, and so Stu bring me some good luck, I guess. And then I identified is Verticillium dahliae. So the first reports in North America being published here. Then just to give you a little bit idea about this genus Verticillium, and there are no sexual stage is found, is known, and but there's more than 10 species being reported. Then I also try to give you this comparison about these the two species. And as I said, the Verticillium dahliae has a broad host range compared to the VL. And then also, if you look under microscope and the short conidia and the VD and the Verticillium dahliae, and then compared to VL. Okay. And so this is the major difference there. So the taxonomy is very confusing, you know, and because many Verticillium species from outside people, from not experienced people, and very similar. So I always consider myself as a plant pathologist, not a mycologist. So I will not get into so much detail. You know, let these mycologists and or the molecular biologists to tell the difference. And however, if I grow the fungus in the plates, and then you can see the colony. And then you notice at the edge of the colony, they will start to produce the sclerotia. Okay. So as I mentioned, the Verticillium dahliae, you will see the microsclerotia is on the plates is a little bit more round, and compared to the one and the VL, which a little bit longer type. This is just to summarize about these two species difference. And microsclerotia is rounded for Verticillium dahliae, conidia a little bit short. This is the range, 3.5 to 5.5 micrometer. And then this is a little bit longer, 7.1 to 8.8. Microsclerotia a little bit longer as well. Then if you look at the filtrates, fluorescence, VL will give you the fluorescence. Then of course the host range is different. The first time I worked on Fusarium Verticillium dahliae was on alfalfa, when I worked on alfalfa disease. Okay, and a very exciting thing happened. And in 2018, finally, there's a way, and because of my stubborn and my persistence. And so I was able to find this first case on this Verticillium and the stripe caused by VL. And as we know, and this is an important disease, and that's why I got CFIA nervous at the first place. And simply because in Northern Europe and Sweden can cause a very big yield loss. And this VL has a limited host range and in fact on the brassica crops, as canola, mustard, all these. And then at the maturity, this VL and can found throughout the plants in roots, in stems, and leaves. Here is the catch. And in fact, I found something and have interest to work on. And I wasn't going to share with you, but now I will let you know my secrets. And I think, of course, I need to do further investigation. And maybe when this Verticillium VL, when during the harvest stage, you will have the infected part. So I think maybe it's on the seed. You know, so I have new projects, want to work on this, can cause the seed-borne disease, or can this seed-to-seedling transmission, you know, because this is a big question for Verticillium. But I still need to do lots of microscopic work and PCR work to confirm it. But the headache part is this microsclerotia of VL, just as similar as club roots, resin spore, can survive in the soil for 10 to 15 years. Then so from this first case we found, and then we see the typical symptom. We see this shredded of skin on the stem of the dead plant, as you can see here. And then we also see the dark unilateral striping on the mature canola plants. And then you also see the microsclerotia here on the dying plants. And then this is formation of the microsclerotia on the microscope at the beginning, as you can see on the right side corner. So I quite often ask ourselves, you know, did we diagnose the Verticillium stripe of canola as blackleg before? Or is there a possible synergistic or additive action together? And so, because sometimes, and when we do the work, and we are working on this Verticillium stripe, but we also can see the blackleg and the symptom there. And then I did a little bit of measurements, and I found, as you can see this picture, on the left side, this is pycnidia, and then on the right side is Verticillium microsclerotia. So they are almost 6 times difference. In other words, pycnidia is 6 times bigger than this microsclerotia of Verticillium. So it's very, you need to use a high magnifying lens to see it, very tiny needle painted, okay? So this is the way to tell the difference. So now I got many people excited with some of the fieldmen and also some of the industry people. They start to send me the specimen last year, 2020, and they promised me they will continue to do so. So I don't mean hopefully, I mean, and maybe there's a chance I will identify more the Verticillium in this year. So I just want to do as a plant pathologist. Number one is important. You have to make sure when you claim you got a VL, it is really the VL causes the canola seedling disease. So we need to complete this disease Cox postulates, the theory. Okay, you found the disease, then you isolate the pathogen, then you try to inoculate the pathogen, and then see you can re-isolate, re-identify. Then you can claim, yes, you found the disease caused by VL. So this is a picture just to show you 7 days after seeding, and there's a check, and then there's a 1X there. That means I use this is the first time study. So I just grow the fungus and in the medium, and then pour in the cups to compare, okay? And has different concentration. And then the bottom picture, you can see there's a control plants, and then this is infected plants. So I completed the pathogenicity test, and I prove it is caused by VL. So I hit the jackpot. We got the VL disease in Alberta. And then this is just to show you the pathogenicity test, the results. As you can see, the control give you better emergency height and also the disease severity 0. But compared to the other 2 isolates, give you higher disease severity and then reduce the height, reduce the emergency. And then if I keep the plants to the longer period time, and then you can see on the left side, the check control start to show the pods and so tall, but then the middle one is VL, so short compared to the Verticillium dahlia on the right side, which tell us this VL, it is more threatening, more virulent than this Verticillium dahlia. And also, you see on the right corner and this upper picture, I have this arrow here. You see, this is the first time I got so excited. This is the first time in the greenhouse I see the similar symptom in the field. Okay, but however, you look at the Verticillium dahlia, don't have this shredded stem. So I was very, very excited to see this, just for my first time eye opener to prove what we read on the paper. Okay, it is really the case. Then I also look at this under the microscope, because you cannot just listen to people. You got to see with your own eyes to prove they are right. The one thing I found very difficult when I work on this fungus grows so slow, okay? And you can see the place here, the medium, the colony is so small, and after 1 month, still so small. So it's not an easy work. Just like the clubroot, but it's still better than clubroot. Clubroot, you cannot capture it at all. But then this one, at least I can capture, but I just need a little bit patience. So you can see the canidiform comparison, the upper part is the Verticillium dahliae, and then the lower side is the Verticillium longisporium. You can see the canidia, it is really a little bit longer compared to the dahliae. And then microsclerotia, a little bit short round. So everything I said earlier, or the paper published earlier, is true. Okay, and so even the symptom, as I mentioned, and on the Verticillium longisporium, you can see this unilateral stripe, and then you can see this shredded of the dead plant tissue. And then for the Verticillium dahliae, mainly is stunting, yellowing, and wilting. You don't see this shredded dead plant tissue. Now we know this has been published in Sweden, in North Europe. They talk about how much yield loss could be 70%, 50%, all these things. And so this is where I got into. As you know, my title as a professor is as applied plant pathology professor at U of A. So it is my interest to do some of the field work. And I'm very weak on this molecular plant pathology part, but the field work is what my passion about. So that's why it's very little known about the verticillium on the yield loss of canola in Canada. So that's why I am very lucky and I'm very grateful. And I got this CARP program, and particularly SaskCanola canola growers, and support my study. And so today I feel I'm a little bit obligated to give a talk today, even I feel in comparison, and maybe I'm a little. If clubroot, you consider me as an adult, but then the verticillium, I'm still doing my child stage, okay? But I will do my best. And so the first study I want to share is last year, despite the COVID condition, and last appreciation to my technician, to all the graduate students, and we were still able to do the 2 sites, field trials. And this is the objective about these field trials. We want to evaluate this verticillium disease severity and at different income to see how the happen to the disease severity. And also we are wishing, like Justine mentioned so much, Canola Council, they care so much. How can we develop a disease assessment scale? And so we need to have more experience in order to reflect what we really see in the field, and then can develop some very practical and easy use scale. And of course, I want to see how this affects on the yield loss, all this. And then eventually, and we can accumulate all these years study together, then we can develop a yield loss model, just like I did and on Blackleg. So the work carried out at the 2 sites, and we use the 2 cultivars, 45H31 CS2000. We have 4 treatments. We use the 3 grain inoculum density, and which is control, and then 100 ml per row, 150 and 200 ml per row. And the plot size is 6 meter times 1.5 meter. We use a split plot design, and then we collect the plants. Then we do the emergency disease severity, yield, all the data collection. And then when we do the field work, as you can see, then you cannot do the conidial suspension. Okay, that's not a particle. So we soak the wheat grain, we autoclave, we inoculate VL, and then we air dry them, then we grind them, we apply in the field. So despite the COVID, we were very productive. So I think the SaskCanola CAP program must be very happy. So this is to show you on June 23rd. This is the result we saw. On the left side is the control. You see the germination, pretty good. And then on the right side is infected with, inoculated with VL, as you can see. So this reduction in germination caused by VL. This is the first picture, okay? And then this is data to back up what you just saw in the picture. As you can see, control has a 100% germination, but if you compare to the high inoculum, will drop to 70% or 83% for 45H31. And one more thing, this is also first time, and I see it in the field. Remember? And in the book, and in the paper, people indicate you will see this leaf yellowing on one side. Bingo, you see here, you only see the leaf and on one side yellowing. Such unique, lovely picture. I'm so happy to see this. And you should see the way I was jumping in the field. Okay, so then about this disease rating scale is a maturity. It's the harvest stage. Then I found out I have this 0 to 4 scale, and the 0 is no symptom, okay, and no pod loss. But 1 is stunting and the discoloration with a little bit darker, but non-pod less. 2 is less than 25% of the main stem colonized by microsclerotia. 3 is up to 75% of the main stem colonization by microsclerotia. And also you can see the stem shredded, plants dead or dying. You probably will question me, how come I don't have a between like a 50% of this? But I want to tell you, in the field condition, it is very hard to catch that level. Okay, at least my first year experience, we will have to see, and the following year, this year, and to prove whether I'm right or not. So this is just to show you again in the picture, like this 0 look like, this 1 look like. Okay, you see this green on one side and the brown on the other side, and then this 2 up to 25%. And then here is about 75% covered. Then this is kind of dead, dying plants. So from this inoculum density effects on the disease severity based on 1 year field trial on 2 sides. As you can see, this disease severity increase as the inoculum increase, low, medium, high, and then go up, and then for 2 cultivars, CS 2000 and also 45H31 at both sides, showing the same kind of trend. And then we look at the seed yield, but this is the seed yield we did per plant. So tremendous work by these graduate students, Ji-Kui. Okay, so again, this is the effects of the disease severity on seed yield loss. Okay, so you can see per plant, of course, as the disease severity increase, and then the yield loss decrease. Again, and for the 45H31, the same thing. So then we also look at the plot yield. We harvest the whole plot. And then once again, you can see the yield loss also kind of reduced, and the yield reduced as the inoculum increased. for both 45, H31, size 1, and the size 2. I'm just because we have a tremendous amount of data. Okay, so time limitation. So I just choose some of the slides to show you, and plus all the slides is very hard, just printed. You know, we are very busy for this talk, and we quickly want to show you some of the results, and in time for this presentation. And hopefully next presentation we can show you more data, more results. And then we look at the mean plot yield. Okay, so we combine these two sides with 45 H31. You can see again this yield. We combine mean plot yield showing the trend, and then we haven't had a chance to really do more data analysis to develop a model or whatever. We just quickly show you this raw data, raw picture. So the conclusion for these 2020 field trials, and I will say inoculate with VL will reduce the seed germination rates at the early stage, and then certainly will reduce plant heights, and the disease severity increased as the inoculum level increase, and yield loss also increase as the inoculum yield loss will increase as the inoculum increase. So after I finish the field trial, and I also want to share with you about the 3 greenhouse experiments we did. And then when we do the greenhouse work, and we realize, and probably is a better idea to do the Canidia suspension. Remember, as you recall my earlier talk, when I did the pathogenicity test, and I just used the agar plug, you know, but really you cannot fix the concentration. And in order to help the breeder in the future to screening all the lines and against VL, so we need to help. That's another major objective in my proposal. And I want to develop the inoculation techniques and what's the best inoculation techniques to screen the lines against VL, and also what the concentration should be, and also what the seedling age should be. So this is the greenhouse study, and I'm going to present to you some of the results. So as you can see here, and we on the left side, and we grow all the seeds, and then so we produce the seedling, and then we pull all the seedling out. And then we grow the fungus in the culture, and then we collect the spores. Then we produce the spore suspension. We go through the filter, several layers of cheesecloth, and then under the microscope, and we do the spore counting, so we know the concentration we are using. So we use the root dipping method, then they're transplanting the seedling into the soil. So this is just to compare again, and with this Verticillium dahliae and the Verticillium longisporum, all at the 10⁷ spore concentration, and on 3 different canola cultivars. And the important thing it is just to show you as this 10⁷ spore concentration per mil, and the VL was more virulent. Okay, so right on, we choose the right pathogen to do the screening. And then this is the second greenhouse study. What we are doing is we use the 2 different spore concentration of VL to do the study, 10⁷, 5 times 10⁷. Then we found out 10⁷ is good enough, okay, to do the test. Although here higher mortality show up with a higher concentration. But I think 1 to 10⁷ is a good concentration for the future screening. It's good. So the greenhouse's third one is to look at the seedling effects on the disease development. So what we did, we used 2 cultivars. We used the CS2000 and the Webstar, and we also use 3 different kind of ages. 1 week old, 2 week old, 3 weeks old seedlings. We use the root dipping methods on this spore and 1 to 10 servings spores per meal for 45 minutes. And then we transplant the plants into each cup. We transplant 4 plants, and we did 15 replicates for each cultivar as each seedling stage. So we do the data collection. Okay. So this is the one, and I want to tell you, do the disease severity scale again. Perhaps we should have 2 sets disease severity scale. I'm trying to inspire the Canola Council, or WCCRC. And so because when you want to do the evaluation, perhaps you cannot just use 1 system. Maybe you need to use 2 systems. And because maybe at the young stage, at the seedling stage, you can use the disease severity from 0 to 6. Okay, if you want to do the breeding against the evaluates against the VL, and then you can do the rating at 10 days after you transplanting. And of course, we do the plant height at 14 days, mortality at 21, 35 days. Then we harvest at 98 days. And then after it reaches the harvest stage, we do the disease severity again, second disease severity at the mature time. So then that time we do a disease score on the 0 to 4 scale. So we collect the yield data by weighing the total seed of each plant. So this is a very interesting results I want to share with you. It's really surprising to me when compared to I did the similar work on these club roots. Totally opposite results. The one I did with the club roots, the one week old, 2 week old, 3 week old, 5 weeks old, all these, and I found out the younger seedling will show more disease severity. But then for this VL study, I got the opposite results. And in fact, we had a meeting at U of A in the greenhouse, and even Steve Strackover, and the professor at U of A also show up. And then Rudolph also show up, the collaborator on these projects with graduate students. Why? How come this happened? Okay, as you can see, this is the control. The first 2 cups from the left side is the control compared to 1 week old, control compared to 2 week old. And obviously, these 2 week old have a more severe disease. It's a total difference from my club root research. So we all standing there and watching the cups and pull our hair, scratch our head. What's the reason? So this is 7 week old White Star, and inoculate the 3 week old seedlings. And you can see beautiful crack on the lower stem, and then the shaded and also covered with the microsclerotia, and then even you can see all the arrow pointed. Okay, so now I think I'm no longer child stage, and I think now I'm young lady stage. I start to build up my confidence. Many thanks for the CARPERS program, the bring me lots of interest on this work. So now, just to show you 7 week old White Star and the 12 week old White Star. Again, you can see the disease severity, and the 1 week old, 2 week old, 3 week old. So the 3 week old and 2 week old showing more damage compared to the 1 week old. And then this is the 98 days after we harvest, and we measure the mean disease severity and about these 2 cultivars. As you can see here, and the 3 week old give you a little bit higher disease severity as you show the pictures. So the results and discussion up to here, okay, about these seedling age effects. we think the symptom increase with age when the plants were inoculated. 3 weeks old seedling, and the disease more severe than 1 week. And then the same thing in terms of the plant height, mortality, and then even the yield loss. So after we had a quick meeting in the greenhouse in front of the plants, and maybe we think the plants 3 week old have a more transplant shock greater than 1 week, or maybe because the 3 weeks old and the seedling have a little bit more roots area exposed to the inoculum when we do the dipping in the spore suspension. And I don't know, and maybe some people, listeners there, maybe you can give me some of your idea, maybe will inspire me to do more work. This is, like I said, this is all the preliminary results we did in the greenhouse. So I want to say thanks to the graduate students, research personnel, and the collaborators. And particularly, I want to thanks to the CARP program, including SaskCanola, Alberta Canola, and the Manitoba Canolas, and the other industry partners support with all the seed, everything I want. And then also thanks to the agronomist and also the agriculture fieldman, and they all call me up and send me, I got lots of specimens there. Okay, and sometimes it's very, very difficult when we do the isolation to tell apart, particular. So lots of work line up, of course, thanks to University of Alberta. So this slide is one of my favorite slides. Canada geese in a row, ready to work. And so I'm ready to work, roll up my sleeves with all the students. And hopefully the COVID will be over soon, so we can then start our greenhouse field work on time. And thanks for your attention. This is the end of my talk. As I said, I'm still at the early stage, and everything I said here is kind of preliminary results, and I'm glad to share with you. Thank you for your attention.
Thank you very much for your presentation, Dr. Hwang.
You're welcome.
Please type your questions into the Q&A box. We will address them after Justine's presentation here during the 30-minute Q&A session at the end of the presentation. Also a reminder, if you are CCA or CCSC member, please send us your full name, CCA or CCSC number in the chat box, and we can get that registered for you. I am now pleased to introduce our third speaker, Justine Cornelsen. Justine is the Agronomy Specialist at Canola Council of Canada. She covers Western Manitoba and Southeastern Saskatchewan. Her lead topics with Canola Council of Canada is Blackleg and Verticillium Stripe management. Justine also chairs the Canadian Blackleg Steering Group. Today, she will provide us with the Blackleg and Verticillium Stripe best management practices. Welcome, Justine.
Great, awesome. Thanks, Keith, for the introduction, and thanks to all three grower organizations for hosting this today. We've covered some pretty in-depth information on both of these diseases, and I'm going to just really take a lot of what they have said already and summarize it down to the field level. And I'm going to start off because I know we've got a lot of agronomists and producers on the call with some questions. So I think there'll be a poll question hopefully pop up on the screen. This will be the first one of the day, so we'll see how smoothly it works. Jeff's working in the background. But yeah, so our first question here. So question number one is, what canola disease is this? And so this poll is going to pop up. We're going to answer question one right now, and we'll see how much you've gathered from the last two presentations. So we've got a few options here. So you can submit your answer, and then we'll move over to the next question. I think Jeff will have to then pop it up again, but we'll see how this works. So here's our question number two. So same thing, asking the same, what canola disease is this? We've got a few different options there. I'm going to note some stem cankering. these are all the things that we actually see in the field and are dealing with. So, right, just training our eye to what we should be looking for, and especially when we're starting to work with new diseases that we're not familiar with. We're still all learning together. So disease ID is becoming extremely, extremely important. All right, moving on to question 3. So we've got a field here, and it's looking at, obviously we can see a lot of premature ripening in the pods going on. This picture was taken in Manitoba the first week in August. So not typically when we see our canola turning this quick, right? So what, we know diseases can cause premature ripening, and looking at this, which disease do you think we are seeing here? So I'm going to end the poll questions, and I'm not sure if Jeff's able to put up our answers for the first 3 or not. But I will let you know in this particular field, this was clubroot, actually. So not a disease we're talking about, but right, it's getting into the field and knowing you're going to be dealing with a bunch of different diseases out there, right? We kind of get our eye trained to maybe just finding blackleg, where we're going to see a lot of these symptoms show up, not only in just the disease realm, when we looked at the fertilizer issues as well, right? There's a lot of things going on. So we've got to take the full story of what's going on in that field into account to make these diagnostic decisions. So in this particular field here, yes, so clubroot patch, which is actually right in front of me. If you were to pull up every plant, they were showing galls. This field also had a lot of blackleg as well. So when you started to look into the cross sections of the plants, you were able to see that internal blackening. So that's what I'm going to cover off today is just getting into the management of blackleg and verticillium. So here is that a verticillium plant showing the premature ripening up in the pods. So you can see this half stem senescence that's going on. Half of this plant is still looking green. The other half is dried down, and we've got pods that have ripened and are actually already shelling out. So that's a symptom that we've always kind of contributed more towards the blackleg side of things, or clubroot, which we are now seeing this in plants that are infected with Verticillium longisporum. So in the next, 20 minutes, try to keep everyone on time today. I'm just going to do a little face-off between the diseases, get into obviously management practices, so crop scouting, so going in there, knowing our diseases and seeing them that way, looking at crop diversity or crop rotation, however you want to capture that. We hear a lot about that importance. And of course, in any disease presentation, I obviously have to put some emphasis on that. And then I'll get into the top 5 management practices that we use for blackleg, and then look at what we know about verticillium and really what we don't know yet. Our management practices are very slim at the moment, mainly because this is a new disease and we are still learning about it. So as for disease face-off, I am going to be covering 2 diseases. If I am sharing them on the same slide together, I'm going to try to keep verticillium on the left-hand side and blackleg on the right-hand side. So we do have the cross-section cuts here to do that comparison. So seeing the verticillium, we do see some haloing. So when we are talking about verticillium, we are talking about the Verticillium longisporum species, which Sheau-Fang did just allude to. This is a soil-borne disease. So it is going to have similar management practices to that of clubroot, right? It is going to move in our soil. It has the potential to move in our waterways, move in the wind, on equipment. So keep that in mind when we're discussing management options. So it does cause the disease Verticillium stripe. I know there's been a lot of confusion in hearing something called wilt. The industry decided on that it's not a wilting disease. We do not get the wilting symptoms like they've seen in regions of Europe. And even over in Europe, they are finding it's more of a vascular disease where they're starting to see the stem stripping. All right, so that outer cell wall is stripping away, and below is where we're seeing the disease. So not so much as a wilt symptom. On the blackleg side of things, right? Blackleg's a disease we're quite familiar with here in Canada. Typically, when we are discussing blackleg, we are talking about the aggressive or the virulent strain of it, or virulent species, which is Leptosphaeria maculans. And blackleg is a stubble-borne disease. So that's why we're concerned with what that old canola residue is doing in the field. And that's why management practices such as rotation really help to minimize this disease impact. And so blackleg, what we're looking for, basal stem constriction, we see that plant lodging, we see the premature ripening as well. And that's where our yield losses are coming from. So as for management, right, we went through a few slides there quick at the beginning, just looking at, what diseases we are dealing with and seeing. Scouting is our number one, one of our number ones, that and crop rotation. So you need to know what you're dealing with. So that's why I put scouting first. You've got to get into your field, know what you're working with. And looking at some of the chat questions already coming up, how did verticillium sneak up on us? It didn't really sneak up on us. It's been there and in the field for probably quite some time here in Manitoba. It went undiagnosed or misdiagnosed, and probably a lot of confusion with that of blackleg, very, very similar symptoms to that of fusarium wilt when we were dealing with that disease. So I think it just got missed for a really long time. And the last few years have been really conclusive for the development of the disease. So it's been there, it's been sitting kind of dormant. We get some symptoms, we typically see them later on in the season. So we're not scouting for this disease, right? We've harvested our canola, we're not going out in the field and looking at that old residue. And that's where verticillium is very, very obvious. So the time of scouting is a little bit different. And this year that window of infection kind of shifted, and that's why we were seeing losses in areas because of this new disease. So going in and properly identifying the disease or your symptoms is extremely important to make future management decisions. When we talk on blackleg, like I said, this is pretty standard. Nothing here has changed too much. Best time to go in and scout and rate for the disease in your field is right prior to swathing or around that 60% seed color change. The reason there is that you're able to still clearly identify the differences between diseases. Once the plants start to die off, it becomes very confusing to know what you're working with. So for blackleg going and scouting, we pull up plants. Ideally, you pull up plants that well represent the field. So you don't want to go and cherry pick the spot that looks really bad. You want to take a representation of your entire field. For diagnostic purposes, that's where I would go to that spot. Okay, what's going on with this patch? But when we're trying to just figure out your overall disease severity and the incidence, taking a really good capture of your field, pulling up random plants, snipping through the root tissue right at the base of the stem. So you still want to go into that root tissue to properly diagnose. And now that I look at this image, I haven't updated the image, but this rating of 0 is way too high. So if you're cutting there, you're missing a lot of that blackleg infection. Where you should be cutting is through that root tissue. And that's where you then can do the rating to determine the level of severity. Here in Canada, Dr. Fernando showed some of the charts. Here in Canada, our disease severity is actually very low for this. Typically, it's around a rating of 1, and some of the new yield loss models that Sheau-Fang's group out of the University of Alberta have shown is that we don't typically start seeing the yield losses from blackleg until about a severity rating of 2. So 50% of that cross section is showing internal blackening. That's where we are seeing the disease or yield loss from this particular disease. So for Verticillium stripe, like I said, new disease, what do we need to be looking for? That's our kind of step number one with our management practices here is like properly identifying it in the field. Lots of different symptoms, and we just need to start training our eye to what to be looking for. Like I said, any of the early symptoms are very difficult to see or not seen in the field. So same, Sheau-Fang showed some really great images of that in the greenhouse where you can see some of that discoloration in the leaves. We haven't been able to really pinpoint that in the field yet. What we are seeing as an early symptom, and what we saw a lot of this summer in Manitoba, was the half stem senescing. So half of that stem still being green, healthy, mature, and the other half starting to prematurely ripen. It almost looks like it's shrunken in and starting to kind of collapse in itself. And I think again, at a normal disease surveying time, it's probably been misdiagnosed with sclerotinia. Sclerotinia will show that type of bleaching. Usually it's all the way around, but depending on the season, I think they would look a lot alike. And where some other confusion has happened. In the stem, same thing, that cross section, it's got that little bit of a grayish hue to it. People think it might be alternaria, might be blackleg. As this progresses and later into the season, you are able to then to start to see the actual mycosclerotia forming in the stem. And that is our really key diagnostic feature when we are looking to verticillium. And here is right again, the stem itself revealing all of those tiny mycosclerotia. So same, if I do the head-to-head comparison between the two, you can see the verticillium stripe, you can see the mycosclerotia, how super tiny they are. And in that cross-section of the plant that's infected with it through the root tissue, you can see that graying across the entire root tissue. You almost see gray, I'll call them specks or flecks throughout that entire. where in comparison to that of the blackleg, you've got more of these black predominant spots that are really kind of chunked together throughout that stem. You're not seeing a discoloration all across the entire root tissue. And on old canola residue, what you will find are the pycnidia or the pseudothecia. They are larger, like they always are described as, black pepper-like. So that's something to note too. They are considerably larger in comparison to that of verticillium. So crop scouting, knowing what we're working with is really step number one. Once you know what you're working with, then we can start to deploy some of these other management options for the diseases. So crop rotation comes next, is one of our top ones for all plant diseases, right? If you're not providing the pathogen with a suitable host for it to do well, it dies off in the field. And this is what we use as a management practice for blackleg. So if you're able to provide a 2-year break, so looking at something like a canola, or I'm from Manitoba, so we use soybean. So canola, wheat, soybean, canola rotation, something like that, allowing that 2-year break from canola allows majority of that residue to break down naturally in the field. You still are going to have some that survive, but you're reducing that overall inoculum level within the field. That being said, I'm sure everyone's well aware of what the canola markets are, and we have been kind of stable at producing, in that range of 20 million acres of canola across Western Canada. We do have fields on tight rotations. When we get into the northern half, right, we've got a lot of fields where it's canola snow, canola wheat, and we are building up our levels. And these are older Ag Canada frequency maps. Since this period when these were done from 2009 to 2014, our acreage has increased. So these maps are still going to look very similar to today anyways. And what they're showing is just the frequency of canola. So anything that's showing red or blue or purple are showing canola either every second year or almost every year. I think the purple dots, are 6 out of 6 years that particular piece of land had canola on them. So I'm, here's Riding Mountain. I always say I'm from there, but I'm actually down here somewhere in Vernon. But you can see that this part of Western Manitoba and Eastern Saskatchewan, what we're seeing here and just the level of canola. So we know we're giving the pathogens the hosts they need to do well in. And same for clubroot management, blackleg management, right? Allowing that at least a 2-year break is going to help reduce that inoculum load. When we talk about verticillium, though, there's a lot of unknown. Same thing, Sheau-Fang had mentioned, the mycosclerotia are going to survive in the soil for the 10 to 15 years. Here in our Canadian conditions, we're not sure at what level they're going to decrease at. Like we know with clubroot, right, we know a population or they're going to die off exponentially. We're not really sure yet. We're starting to wonder if the mycosclerotia are a little hardy and they can withstand and survive longer in the soil. And European work is showing, right, that longevity. So we don't have a cropping rotation message within specifically for verticillium, just overall that, you are going to be reducing inoculum. If you're not applying a host crop, you're not able to allow that disease to build up. So verticillium is a brassica specific, or Verticillium longisporum is a brassica specific disease. So we don't have to worry about it showing up in our cereal crops. I just wanted to highlight some of the great work that's been done here in Canada on cropping rotations. This was some of Neil Harker's work looking at the effect of blackleg incidents. And what it's showing here is, this is, 3 years of continuous canola. So you're going to start off with a little bit of a lower blackleg incidence. As you increase every year, you're going to start to move that bar up higher and higher. So 6 years of continuous canola, you're starting off with 60% incidence. And every year you can extend that rotation out. You're seeing the inoculum, that blackleg disease, die off within the field. So like I said, we put a lot of emphasis on crop rotation and minimizing the hosts or susceptible hosts. So because we don't know a lot about verticillium, I'm going to focus on the management practices for that of blackleg. These have been, well defined. We've been using the same stuff for quite some time here in Canada. There's been some new changes or some new additions recently to the list. But like I said, we've covered off crop rotation and the importance of scouting and properly identifying the disease. And then I'm going to just touch upon the use of resistant hybrids, looking at the rotation of resistant groups and resistant hybrids and the genetics behind that, and then look at some fungicide products. So Dr. Fernando had covered off kind of the situation. Everything we grow here in Canada is a resistant hybrid. One of my trick poll questions, which I wasn't going to pull up, was, are you growing a resistant hybrid? And yes, everything on the market today is either resistant or moderately resistant to blackleg. And this is just to showcase, why we do this and how severe it is without growing resistance. So this is Westar. This is the check that we use. This has no blackleg resistance, so no quantitative resistance, no major genes, nothing. And this is what we would be dealing with under pressure, so blackleg pressure. So completely flat crops. And I know some of the growers here on the call probably remember growing Westar, right? We've come quite a few years since then, and now we are growing resistant hybrids. So same, I won't cover this, the field resistance, right? Everything on the market is resistant to blackleg. But it's now we're starting to see these changes. So saying this is a resistant hybrid. Why are we seeing so much disease in it, right? We've got some fields that are at some really elevated incident numbers. So a lot of plants within the field showing it. And we do have fields that are getting yield loss, so over that rating of 2. So what can we do about it? And what the one suggestion was picking your hybrids based on the genetic information. So the information that we know we want producers to have and be able to make decisions based on that. So this gets into the big blackleg resistance debate. And I know there's quite a few questions already in the chat looking at, quantitative resistance and how do we use that and how do we measure that. As of right now, what quantitative resistance is, it's broad and it's non-race specific. So when you're growing something that has quantitative resistance in the field, it is going to work towards all the races of blackleg that are out there. However, that being said, it still allows the pathogen in, and this is what this graphic is showing. It is not a complete defense. So it doesn't, the plant doesn't initiate this defense response that stops it. It allows the pathogen in. And how quantitative resistance works, it works at slowing that rate of infection. So it might allow the pathogen in, but it's not allowing it to spread and cause these really severe disease symptoms, which would be contributing to yield loss. Once again, however, that being said, we do, under severe pressures, you see quantitative resistance start to fail. It can't withstand high inoculum levels. So if you continue to grow canola in tight rotations, you continue to have that inoculum rise in the field, the pathogen is able to overcome the genes that are being thrown at it. So even that quantitative resistance starts to break down. As of right now, quantitative resistance, we have no way of measuring it. So I saw that comment time and time again. That's something that Dr. Fernando had highlighted some of the research projects that are looking at quantitative resistance. This is not just a canola issue. This is a pathology issue, right? This is not just for canola. This is all crops. We don't have a good way of measuring quantitative resistance because it is made up of so many different genes working at different points in that plant's life development. We can't really pinpoint exactly what's going on yet. So it's very, very complex. So that is why there has been a focus on understanding the major genes. And I shouldn't even say understanding. We know a lot about the major genes. So it's taking that information and doing something about it. So the way the major genes work are right there, they are race specific. So they have to match up to different races within the field for them to initiate that defense response within the plant. If there is no match, it does not work. And that is what we're seeing. We've been using a gene that no longer matches the predominant race profile out there in the field. So we're not seeing that defense response kick in. And so the hybrids are now relying on that quantitative background. So in a perfect world here, what we're wanting is the combination. You want a major gene in your hybrid that is going to match the predominant races, backed up by this really fantastic quantitative background, which is going to work at varying levels or varying degrees towards different races within the field. And Australian work is showing that too, that the quantitative resistance is actually reacting differently to different races as well. So it's not as broad spectrum as we had hoped. They are seeing differences. So the one new thing with the industry is with this introduction of the major gene labels. So there's a lot of interactions and different things, and that's why they are placed in groups. So that's why you're not seeing just an RLM3 labeling. A lot of companies, when they do label, they do put both for information, which I think is valuable, right? The more information we have, the more well-informed we are to make decisions. So we do see resistance gene groups broken up that way. So I'm not going to get into a whole lot of detail there. On your canola seed bags, you are just going to be seeing typically that R rating or MR rating on the bag. You will have to go into your seed guides to look then for the major gene labels. And typically companies do present them all together. So if you look at, I'll use Cargill's example. If you look at the Cargill lineup, they show that which genes they are using, the hybrids, they put in their clubroot genetics, you name it, it's all there in one graphic to look at. So like I said, this information complex. Dilantha had talked about, we're able to test our stubble. So taking old canola residue to figure out the races. From that information, it is very complex still, but you can figure out which major gene hybrid to then grow next or to use in rotation on your farm. So why the industry moved this way, I think it's really important to understand, right? It's not a silver bullet. It still needs to be used in an overall IPM approach or an integrated pest management approach. What it does, it preserves our genetics. And we can learn a lot of what's happened in the clubroot situation, right? Within a very short window of time, we've been able to burn through a lot of our resistant hybrids and the genes being deployed. Blackleg is a little slower moving, so we're not seeing that aggressiveness, but it's coming. And so what we want to do is preserve these genetics so that we can actually use them in the long term. So finding out ways to properly steward them. And because we're not just coming up, life science companies are not developing new magical genes or new resistant hybrids every year, right? It's something that they rely on those tools that are in place. And now we want to continue their longevity. So using them in an overall approach will help reduce the overall disease pressure, along with things like proper scouting, crop rotation, and just overall use of a resistant hybrid. To the quantitative point, like I said, we're working on it as an industry to understand further. But as of right now, when we see quantitative labeling or that a hybrid has great quantitative resistance, there's no way to measure the difference between hybrids right now. It's not, there's no industry standard there. There's no scale there right now. And in a lot of the work, and like I said, Dr. Fernando had touched upon the work that Dr. Pang's doing and Dr. Borhan are doing. What's going on there is that you need to know what that major gene is first to then measure that quantitative background within the hybrid. Okay, switching gears. Like I said, we've heard enough about resistant hybrids. Fungicide use. So this is another newer area. I guess not this part, but for the longest time we've had the option to apply a foliar fungicide. It is not a common practice out there. This is a preventative practice, so you need to put it on or spray your canola before the disease is attacking or before the pathogen is attacking. And this is something that we just don't get on early enough to protect the plants. Jug recommendation for any of these products for a foliar fungicide application for blackleg are that 2 to 6 range. The critical window for blackleg infection and where you're seeing yield loss is really from cotyledon to 2-leaf. So you need to get on extremely early and coat those plants before the disease or before that pathogen is trying to attack. And so Dr. Pang has done some work on this and just showing there really was no benefit from applying a fungicide application when your resistant hybrid is functioning to full capacity. So if it's not acting like a resistant hybrid, if its major gene is being overcome, that's where an application would pay off. And really what you're seeing is you're just reducing that overall incidence. So if you continue to grow canola on tighter rotations, if you are using a fungicide application, that is going to reduce some of that inoculum that's building up over the years. That being said, there's also a fungicide sensitivity component to think about. And some of the work out of the University of Alberta is started to highlight that there is this potential for some sensitivity where the product is not going to be managing the disease. The one new part is fungicide seed treatments. So blackleg right now, when you buy, or canola when you buy right now, is seed treated with a fungicide, and what that is protecting it against is airborne spores or seed transmission, which is no longer a risk because we coat everything with a fungicide. So there's no risk of transferring blackleg within your canola hybrid bags. or from field to field that way because the fungicide covers it then. We don't have any protection in field from that fungicide. So once that plant starts growing, the seed treatment isn't taken up until now. We do have products. So this will be the first year where canola growers can access or buy some products that will be a seed treatment to protect the plants when they're growing. So they'll be systemic, be very similar to that of flea beetle treatments. So once that plant starts growing, it takes in that seed treatment and it is protected early on from the pathogen entering it. So looking at this example, this is work out of the University of Manitoba, and this is testing a Syngenta product. So this will be the product, Sultra will be the product that is available this year. What it's doing is it is protecting early on. So this is cotyledon stage, and this is Westar, our susceptible, which shows blackleg all the time. Sultra is now able to protect it. And a lot of this work, and Dr. Gary Pang has done similar work on other products as well, or other seed treatment products for blackleg. And what happens is you find a product like this pay off once again when your resistant hybrid is not functioning, when that major gene is not matching that predominant race within the field. So this is where a product like that would hopefully solve and also minimize or reduce that opportunity of trying to get in there with a foliar fungicide, right? Because we missed the timing, something like this is actually hitting the timing. So a seed treatment would kind of outweigh the timing and the efficacy of a foliar fungicide because it is protecting when that critical window of infection for blackleg is occurring. Like I said, that's our blackleg. That was kind of a quick run through of them. Just for time's sake, I won't eat up any more time on blackleg. We're familiar with the list, and a lot of this is on blackleg.ca. So same thing, back to just a few verticillium slides, looking at it. Here's once again the cross sections. I'm just going to keep ingraining this in our minds of what to be looking for, especially as we move into next season, the symptomology that we've been able to identify for verticillium. So right, the grayish hue across the cross section in the root tissue versus the black predominant spots that are seen for blackleg. And then to really mess with everyone's minds, you will find both of these diseases coexisting, or both of these pathogens coexisting. So I swapped my images here, but yeah, verticillium would be the top piece here, right? So the teeny little specks all across, and blackleg here would be the larger black predominant spots. So the pycnidia forming on the stem. So as for verticillium management, it is the unknown right now, right? New disease here in Canada. We've taken a lot of work and research out of Europe, but even there, they're still struggling to manage this particular disease. So to date, right now, there's nothing on the market here in Canada that would be preventative or curative in the form of using a fungicide. So no seed treatments, no foliar applications for this disease. One management practice, which is kind of downplayed a lot of the times, but it's just the controlling of brassica weeds or canola volunteers, right? Weed management is a foundation component of any management plan, right, or any disease management plan. So if you can get rid of the host, you're not going to allow that inoculum to build. We are seeing susceptibility between different hybrids. So same, Dr. Sheau-Fang Hwang's work was showing that, right? We are seeing the differences in susceptibility. I know being in hybrid trials, you can see it out in the field. So we are looking at that. There's lots of work over in Europe trying to identify the genes that are at play here. So same, this will be something down the road. We will likely have hybrids that will be showing resistance or tolerance towards Verticillium stripe. Lengthening and rotation, I've mentioned that. Because this is a soil-borne, right? This is where it becomes very complex to manage. The nature of this disease, and when we're going in and harvesting plants, right, we are cutting through that stem tissue where the microscrotia are being held. So they have that very easy capability of spreading on our combine, in the dirt, in the wind. And in a lot of the fields here in Manitoba, we typically are seeing the disease widespread. We're seeing it across the fields, plants kind of scattered throughout. So we know it can easily spread. We found it all across well, and the environmental conditions have been very conclusive for disease development. I'll move over our Blackleg because I said I know we are running to the end of this presentation, but lots of resources that are captured here. A lot of the research that you've heard today will be summarized within the Research Hub. A lot of the final reports end up there, along with on the grower organization's research pages as well. And if you want it in the easy to digest format, Canola Watch is the best place to find it. I know I rely heavily on Canola Watch to kind of get to the short, the sweet, and to the point on how to manage and what to be looking for a lot of these diseases. So with that, I think we do have a few, or are moving into some question periods here. If you have any further questions for myself that can't be answered in the question period, feel free to reach out at any time, and I'd love to talk further on these two diseases.
Thank you, Justine, very much for your presentation. So as Justina mentioned, that we are heading into our Q&A portion of the meeting. It was set to wrap up at 10 o'clock. I see we're very close to that, but we will extend that out here for some of the questions that haven't been answered. Justine did a great job of answering some of the questions that were posted here earlier. So if you have, your questions can be asked by typing into the Q&A box, and they will be addressed as they come forward. So if you have specific presenters that you wish to hear from, I ask that you please address those in that question. So going through, and we had lights for to try and set priorities on the questions. So the one with the most was about the minor gene resistance and how you qualify that. And I think, Justine, you addressed that very well, that there's really no good way to do that. There's how is the effectiveness of a rotation of major genes measured if the hybrids have minor gene resistance? Seems like a confounding factor. No minor R gene resistance. Justine, you addressed that some, but did you? Any more comments on that, or Dr. Fernando?
Yeah, I think if I can take it for a quick second, and then I'm sure Dr. Fernando can really elaborate on that. It's understanding how these two different disease or resistances work, right? So the major gene is the first line of defense. And what it does is when the pathogen is attacking, it stops it right there and then. So quantitative never gets the chance to even play if major genes work effectively and are matching the races. It's when that major gene is not working, the pathogen enters in, and that's where the quantitative resistance comes into play. And then it will work at slowing it down. So that's how we are able to measure the effectiveness of major genes, because they are the first line of response. If the pathogen can get past them, then it's up to the quantitative resistance. I don't know if Dr. Fernando wants to add to that, but
I think Justine answered it so well. I don't think I have anything else to add. I think it's a very good question, but I think also exactly what Justine said really is how it happens. So there's no other way to say it better.
Thank you. Jeff, could we, I know the meeting was scheduled to end at 10 o'clock, so we might have some people that need to leave, and we're maybe looking for that QR code. So I was wondering if you could put that up on the screen here just for a little bit, that we can, that people can scan that in if they have that app. Thank you.
You bet.
So there's a question from Ron, and you addressed this to somewhat, Justine. Why this season was so devastating with the verticillium when we haven't seen symptoms years prior in our valley? And talking to other producers, the disease was evident over a majority of the area. So if you comments towards that, please.
Yeah, and I'm guessing when we're referring to the valley, I'm thinking Red River Valley, and that's where one of the first cases, the first case of Verticillium longisporum was documented, right, just outside of Winnipeg. So like I said, we've known it's been here for quite a few years, and I really think it's been the environmental conditions why we've been seeing it really take off the last few seasons. We've probably just not been diagnosing it. A lot of the first fields that were diagnosed with verticillium were found later on in the season. And when I refer to later on in the season, I'm talking, you know, a few weeks after harvest is where you can see the really evident signs of it. In season, we just haven't been seeing the field symptoms to raise any alarm bells to think that there's an issue. So with these last few dry years, we've allowed the pathogen inoculum to build in the field. And I think that's why in 2020, the conditions were just really right. We had a bit of delayed seeding and kind of pushed our harvest window back a little bit. And that's why we were seeing some of these extreme symptoms into mid-August and into early September this year.
Thank you. Next question was, any correlation between finding verticillium and the adaptation of straight cutting canola versus swath?
So great question. I know it's something that was on a lot of our minds. Because this is a stem or a vascular disease, right, and it's moving up into the plant from the soil into the stem, the longer the plant is out there to mature, right, the longer it's at risk. So, right, with a lot of those straight cut hybrids, right, you are leaving them to full maturity. So they are pushed back later into September anyways. And it's allowed that pathogen to move in and cause more damage. So there's no direct correlation yet. There's nothing backed by research, but that is something that we will be looking at further and potentially why we're starting to see it more often. I get lots of claims that people have seen this disease, 10, 20 years ago. And we've now shifted towards more straight cut acres. So right, there is that potential of building that inoculum because we're allowing that pathogen into the plants, and it just keeps up building. The later it in, the more infection that pathogen is able to cause.
Thank you. So will Cilantro help defend the plant when flea beetle feeding is high? We can see infection severity increase with feeding. Anybody want to take that one?
I think Justin would have something to say too, but I just wanted to let you know that because we worked on the Saltro, one of my PhD students worked on the Saltro, the way that the pathogen, one of the things that we also did was to use a fungus, blackleg fungus that had a green fluorescent protein so that we can monitor the fungus within the plant. And what we saw even with Vesta was that the pathogen was not moving into the plant, into the cotyledon. So I think the way to answer that question, it's a great question. We haven't tested it together, but the flea beetle may not have much of an impact, even though it has caused the flea beetle damage itself will be there. That I don't think that would change. But the Saltro has stopped the damage that can be caused with that interaction of blackleg and the flea beetle by cutting off the blackleg altogether. So that's where I think the answer should be going. And I think it's a great question. So because we are working on the flea beetle aspect as well in my lab with my postdoc, so I think this is a question that we can answer in the years to come next year, maybe with some testing, real testing done with the two.
Thank you. So what percentage of current varieties are labeled for the blackleg resistance now?
So on the market today, there would be roughly half of life science companies that are identifying their major genes. That's not broken down by market share by any means. We've got two of the larger players still not identifying their major genes yet. So a lot of the smaller players are, and like I said, this information can be found through the seed guide and also looking at companies' hybrid lineups. And that's where you'll find the major gene info.
Just trying to find the next one. Okay. So was the spike in I believe this is your presentation, Dr. Fernando, was the spike in the 2013-14 due to higher moisture conditions? So talking about one of the slides, so So maybe I'll also have you refer to, like, what is the effect of moisture? Like, it seems to vary across the prairies from year to year and within provinces. So how much effect does moisture have on that variance of blackleg from year to year?
Yeah, excellent question. And I think I should have addressed that in my presentation as well. If you look at the graph again, you will see that Saskatchewan has much lower disease, even though it is now progressing up. You can see that that is because of the lower levels of moisture compared to Alberta and Manitoba. And Manitoba in particular has had a fairly significant role to play with blackleg, mainly because of the moisture levels that are fairly high. So it really correlates with the moisture that is available. So I don't have the data for 2013-14 in my mind right now, but most likely if there was a higher increase, there would have been a good correlation with the moisture at that time. Keith, can I take another question that I think is very important to answer? I don't know the person who asked, but it's an excellent question, asking what soil should he or she send to the testing laboratories for blackleg testing. So I should emphasize that it is not soil, it is the stubble that is needed to be sent. So what you look for is the stubble that is infected in your field. Let's say you were a grower of canola in 2020 and you had blackleg fairly severe in your field. So you collect your stubble randomly. Again, when I say randomly, you are collecting disease stubble, but from different areas in your field, so that you are making sure that it is not a clustered particular race structure that you are going to get as an answer. So go through the field, collect some stubble, and then send it to the places like 2020 or Discovery Seeds or PSI, and they will do the race structure and give you the result as to what your population is.
Thank you, Dr. Fernando, for addressing that one. And that sort of leads into the next one. The 4-year testing is short. So what is the potential for the pathogen shift within a field over a longer time period?
Can you repeat that question one more time, Keith?
So it says 4-year testing is short. So what is the potential for pathogen shift within a field over a longer time period?
Okay, so if I understood the question correctly, actually that work has already been done in my lab by another PhD student. The publication came out in December, so I will share it with the Canola Council of Canada so that they can make some of those that information available. But to make that available to this audience, rotation, even a very short time, the virulence of a particular isolate can change from avirulent to virulent. So when, as Justine mentioned, when it is avirulent, it's a perfect match with the R gene, so there is no disease. As soon as it changes to the virulence, then there is disease. Now what we are understanding is, now this needs to be tested more. It seems like there are certain avirulence genes, when combating with the R gene, changes faster, and some may take a longer period. So there seems to be certain stability in certain of the pathogen genes and certain that are much more volatile and change faster. So it's a great question. All that I can say is that research has been done, and we have been able to show that the pathogen can change even within a year. So that's not a good news.
Okay. Thank you. Just trying to pick through the next ones here. Okay, so at what stage was verticillium severity rated in the canola field? Field symptoms appear to advance quickly, so a standard assessment timing would be critical. Would you agree? I'm not. Um,
I think that's a question for Sheau-Fang. And I'll let her elaborate if she's still on. If she's not, I'll kind of keep going. It's something that the canola industry is working on developing an industry-wide disease rating scale. So that's something that we're going to vote on here within the next few weeks to have in place. There's a bunch of different models, and some of the rating scales have used wilting symptoms, which, like I said, we struggle to find here. And some of Sheau-Fang's work, right, via in the field versus the greenhouse or the growth chambers, they're using different scales as well. So something that they're using in the lab isn't going to necessarily work out in the field. And where we want to be looking for disease and why we rate canola diseases at that 60% seed color changes, so we can determine what the yield loss is. So we would be looking for something to measure then, potentially a little later in the season. But once again, the later infection isn't correlating towards yield loss. It's just building the inoculum up. So this is why it's a little difficult and why it's probably been missed, because we just don't see it that evident at 60% seed color change or right prior to swathing.
Thank you. So any research being done on caramba application at herbicide timing for action against blackleg or verticillium? Caramba is a known growth regulator at the application timing for canola.
We haven't done anything. Maybe Sheau-Fang, I don't know whether you have done. I have worked with Blackleg and Verticillium, but we haven't done that kind of fungicide testing.
No, and with caramba being a plant growth regulator, I believe is the technical term there. A lot of those things, right? Any sort of application, if it's applied at the herbicide timing and you're not applying early, you're going to miss that critical window for blackleg infection. And like I said, the products that we have work well, but they're not applied early enough. And like I said, it's a very small percentage of growers that actually do apply a foliar fungicide to manage or to hopefully manage blackleg, but they're missing that window. So the earlier, the better. And same. So if you're going in at a 4-leaf time, a lot of that infection has already come in and through. And once the plants are infected, you can't reverse what's been done. So you need to coat it before. And that's how our fungicide products work. And I'm not up to date on my caramba knowledge, but the same. I think everything's just applied a little too late.
Thank you. I think this next question was directed to Dr. Hwang. So I don't see much stunting at early stage in the field from Verticillium stripe. Symptoms usually show up in the summer. Why the difference from growth room work?
Is Sheau-Fang no longer with us on the call?
I don't think she is. No.
Okay.
No, she's she's
okay.
She's
she
gone.
is gone. Okay. To address, I guess that question, like I said, we do see the disease perform differently, and is it maybe because they're working in single pots, controlled conditions, right? Maintaining a soil temperature. A lot of the work in Europe is showing that really close correlation to the soil temperature and how this disease is able to infect plants. So I think that's why within the lab setting we're able to see that early wilting symptoms, where in the field landscape we just don't have the conditions that are conclusive to be showing those types of symptoms yet.
Okay, I think that that's all the questions that were in the Q&A. There was a really good question that was in the chat, and I didn't see it come back up in the Q&A again. I'm just trying to see if I could find it, but I mean, if I don't get to it here, so all of your email addresses have been posted up, and the session is being recorded. So all the people who are registered will be sent a link, and you'll be able to go back and find those email addresses, and also just view the presentations again and through the question period. So as Mickey, has that got all the questions? I might have missed one or two. Have you been keeping track, please?
I think at this point, we will encourage people to directly contact with the speakers to address their questions, Keith.
Yeah, okay, thank you very much. So, and thank you very much for everybody participating in this morning's webinar. And thanks once again to all of our presenters. Please join us for the next webinar on club root management, which is scheduled for February the 11th. The registration page for the other webinars can be found at SaskCanola, Alberta Canola, and Manitoba Canola Growers websites. So thank you very much.